Thank you for joining us for Rappaport Connect's Alumni Spotlights, where we interview notable Rappaport Foundation alumni. Today, we are joined by 2023 Rappaport Research Fellow, Dr. Rebecca Gillani. Dr. Gillani is a physician scientist in the MGH division of neuroimmunology and neuroinfectious disease. She is a clinical expert in multiple sclerosis and an instructor in neurology at Harvard Medical School. Rebecca is here to discuss her research on how inflammation damages synapses in an experimental model of multiple sclerosis. Rebecca, thank you for joining us today. Why don't we get started? First and foremost, let's start with the basics. Can you give our viewers an overview of multiple sclerosis and how it impacts the body?
Sean, thanks for having me today to talk about our research. I'm really excited to be here. Multiple sclerosis is an autoimmune condition that impacts the brain and the spinal cord. And when inflammation attacks the brain and the spinal cord, this strips away the protective coating around nerve fibers. That's called myelin. And this leads to attacks of neurologic symptoms such as weakness or trouble walking.
What are neurons in synapses and what role do they play in brain function?
Yeah, neurons are the cells in the brain that do information processing that allow us to do all the things we do with our bodies walking, talking, thinking. And it's hard to believe, but we all have billions of brain cells or neurons that serve all these functions. And they're connected together with these connections called synapses. So those brain cells are constantly active and talking to one another through synapses.
Rebecca, let's talk about your research. What question did you set out to answer regarding MS and synapses?
Yeah, to answer that question, I first want to tell you a little bit about the landscape in multiple sclerosis treatments currently. So, we have a multitude of medications called disease modifying therapies that can do a good job at preventing new spots of inflammation in the brain and spinal cord and new attacks of neurologic symptoms in people with multiple sclerosis. But one of the challenges is that despite treatment with these medications, some people with multiple sclerosis continue to have gradual worsening of their neurologic disability. And also, the majority of people living with multiple sclerosis have cognitive or thinking problems. And so, the big question is, what are we missing? What is it that needs to be treated to try to prevent that gradual worsening of disability and these cognitive problems in people living with multiple sclerosis? So, I think one of the pieces that we're missing is that we now know that there's really widespread damage to neurons and synapses, so brain cells and their connections, in people living with multiple sclerosis, even outside of those discrete areas of inflammation. So it seems that we need to target this damage as well in order to treat these additional symptoms.
Rebecca, what methods did you use to conduct your research?
What we do is we use experimental models to model the inflammation similar to what happens in someone living with multiple sclerosis. And in these particular experiments, we wanted to ask the question of let's actually look at neurons and synapses and see what happens to them when they're exposed to inflammation. So, to do that, we use methods to fluorescently label neurons and their synapses. So, basically, make them light up with a color. And then we use a sophisticated microscope that allows us to take pictures of these neurons and synapses in this experimental model as they're being exposed to inflammation.
And Rebecca, what have you found in your research thus far?
In these experiments, we wanted to look at how synapses are shaped by inflammation. And what we found is that synapses are destabilized by inflammation. So, in the healthy brain, synapses are actually not fixed structures. They're constantly changing as we learn, as we're exposed to the environment. However, in the setting of inflammation, this process of dynamic changes in synapses is accelerated. So they're coming and going more quickly and are destabilized by inflammation. And we found that this is not just targeted to small areas. This is really a widespread problem that's affecting the whole neuron. So inflammation is widely destructive to neuronal function. But the really surprising thing that we found was that despite this instability and damage that's being done to synapses, when we looked at these synapses using a super resolution imaging technique, we saw that their structure actually appeared to be normal. And we didn't actually see a large loss of synapses. There was just a modest loss of synapses. So I think this is actually a little bit of a silver lining or a positive thing, and that it shows that while inflammation is destructive and destabilizing to synapses, the brain is still able to form normal appearing synapses, and there's not large loss of synapses, at least in the setting of early neuroinflammation. So that seems to be an opportunity where if we, in the right setting or with the right treatment, we might be able to protect those synapses.
Fascinating. And what's the next phase of your study?
Yeah, so I think there's two really important next steps. The first one is to ask what are the drivers of this neuronal damage in the setting of inflammation? And that's obviously very important because if we know what the drivers are, then we can design therapies to try to target these pathways and protect neurons that are being exposed to inflammation. And number two is we want to ask about what is the functional consequence of this neuronal damage? How does this synaptic instability change the function of the neuron? And then, of course, as a consequence of that, the cognitive functioning.
Well, we look forward to hearing about what comes next for you. Rebecca, you are a 2023 Rappaport Research Fellow. Tell us about how you learned about the fellowship and its impact on your research.
Of course. So I learned of the fellowship through my colleagues who have been fellows in the past, including one of my close collaborators who worked on this project with me, Brian Baxke. And the impact of this fellowship really goes towards helping to support people early in their career. So, both for him and for me, this fellowship has been instrumental in helping to launch our research careers, which then pays dividends for years to come as we're going to continue this work to better understand neurological diseases and develop new treatment strategies.
It's wonderful that you're collaborating with Brian. He was a fellow between 2012 and 2017. How did the two of you come to collaborate on this?
So it started quite a few years ago. I was a postdoctoral fellow at MIT studying synapses in the healthy brain. At the same time, I'm a multiple sclerosis neurologist, so I care for people living with multiple sclerosis. So when I was doing this research as a postdoctoral fellow, I started to think about how does inflammation damage synapses and started to conceive of these ideas in this project. And I asked, how can I put this into fruition? So I started looking around. And Brian is a world renowned expert in studying the brain in the setting of neurological disease using advanced imaging techniques. So he primarily studies Alzheimer's disease. So I reached out to Brian and he agreed to meet with me. And then it's sort of been history from there, and that we agreed that we shared common interests and we decided to collaborate on this project. And now I'm happy that it's come to fruition and that we're going to be continuing as collaborators for years to come.
That's wonderful. Rebecca, thank you so much for your time today, and thank you for enlightening us on your research. It's been really terrific having you here.
Thank you for having me.